You all for coming to this evening's talk on Glacier Biogeochemistry. I'm Martyn Tranter, and I'll be handing you over to Mark Skidmore and Jon Hawkings for something much more interesting than I've got to tell you. The outline of the talk is I'll introduce some of the basic physical and biogeochemical processes under ice before handing over to Mark for subglacial iron, sulfur and cycling and sulfur and carbon cycling. And then Jon will talk about ice sheet fertilization before you come back to me for something that I'm quite excited about, a new turn in the field. I want to start off with our acknowledgements. We've worked with tons of people. Our supervisors have been really influential. Our students have been fantastic. They're too numerous to mention, but this talk is more for them than it is for us. And our funding agencies are shown by the logos below. So when I started my PhD back in 1978, all I knew about glacial erosion was that, as far as I was concerned, it crushed up silicate bedrock to liberate the trace carbonate and sulfide minerals that are present in most silicate rocks and bedrock, rocks and bedrocks. And carbonates and sulfates, they're really reactive compared to the boring old silicate rock that they're contained within. What I went on to learn then was that glacial erosion is the biggest producer of silt-sized debris on the planet, and just about every glacial runoff stream is turbid, and it contains silt-sized debris. If you look on the backs of the silt-sized particles, you always find microparticles. And I didn't quite appreciate back in the day that the rock will always want to react with water. So glacial flow will always try to react with water if it possibly can. And then under the glacier, then the particles are wetted at least periodically. If they're not water transported, then periodically they get bathed by regelation water. And sometimes they're bathed by freeze-on waters at the bed as well. And what do I mean by these microparticles? Jon will tell you more about that in his part of the talk. But the scale on these slides, this is 200 nanometers, 0.2 of a micron. A clay-sized particle is about 2 microns or less. And you'll see on the back of this silicate particle, there are little microparticles held electrostatically to the surface. And these have tons of excess surface free energy, and they're much more soluble than the parent grain. And then scattered in glacial debris is frequently iron nanoclusters, and Jon will tell you more about this later. And when I started my my postdoc work, when I was first a lecturer back in the early 80s, I had the privilege to work on whole Glacier de Rolla with workers from Southampton and from Cambridge. And this, what Julian Dodgwell calls a piddly little lump of warm ice in the Alps. It's only a few kilometers long. It's only a couple of kilometers wide. And the great thing is that the processes that we found at Arolla, you can export, allowing for scale, even to the Greenland and the Antarctic ice sheets. And that points to the ubiquitous nature of a lot of the glacial processes of interest to us. And here's a slide of Arolla back in the early 80s. Since then, it's retreated round the bend by several hundred meters. It's a sad place. And I was fascinated by this turbid runoff coming out of the glacier front. And I fell in scientific love with the diurnal discharge cycle of an alpine glacier. It always gets me going. You know, the sun comes out in the day and the discharge rises. It goes to bed at night and the discharge falls. And then inverse to that, low discharge waters have high conductivity. They got lots of salt in them. High discharge waters have low conductivity. They haven't got much salt in them. And the idea is that there must be two components to glacial runoff, a dilute component that flows somewhere along the surface. It was thought back in the day, and a subglacial component, something that flows along the bed and that picks up the solid from the weathered rock floor at the bed. Even worse, an annual hydrograph of an Alpine glacier, I just see tons and tons of things in there. And I can give lectures on this to undergraduates who've been bored silly by me talking about it. But the main thing is high discharge, low conductivity, low discharge, high conductivity. What happens on a diurnal basis happens on a seasonal basis. And I suppose my contribution to the subject was stitching the chemistry onto the conductivity and the discharge changes. And our holy grail was to show that there were one set of chemical weathering reactions going on in the channelized drainage system, and there was another set of reactions going on in the distributed type drainage system. Distributed type drainage systems are high pressure, channelized type drainage systems are low pressure. We made some inferences about the types of reactions we thought were going on at the bed, and NERC very kindly funded us in the early 90s to go back to Arolla and to drill through 100 meters or so of ice with a hot water drill for the first time, to get down to the bed and sample the waters to see if we'd got the reactions right. And I was working at the oceanography department in Southampton, and I nicked the idea of a Niskin bottle applied to a little hot water hole going through the drill, going down through the bed, and used Bryn Hubbard with the first subglacial meltwater sample that we collected back in the early 90s. And if you think you're going to find what you're looking for, you're often completely wrong in science. And here's something that we found, and it's only 1999 that this came out. So a little over 20 years ago, people didn't think that glacier beds were alive. Now we know now we know that they are. And these were the sort of diagrams we hand drew back in the day. This just shows sediment concentration logarithm. This is the number of microbial cells we found. Wow, and that they're divided means that they're happy growing cells. So we found life at the glacier bed, and that was very helpful for me because it helped me understand the types of weathering reactions that occurred at the bed. And the big reaction at the bed is pyrite oxidation. Fool's gold gets oxidized into sulfuric acid, which dissolves the bedrock, and the microbes catalyze this reaction. The wacky thing is there's so much sulfate at the bed, you can't possibly only be using oxygen to oxidize the sulfide. There's got to be other oxidizing agents, and the flow paths go anoxic. We were able to show that there's microbial oxidation of organic matter. We were able to speculate that there was even sulfate reduction going along on some of the flow paths. So the reactions that we found at the bed were sulfide oxidation, oxidation of organic carbon. We found the formation of rust. I didn't realize how important that was. We found that oxygen was driven down along certain flow paths. We speculated on whether there was sulfate reduction. We speculated on whether even methanogenesis could occur, and that seems very likely under the Greenland and Antarctic ice sheets. And we suggested, to make the connection to the earth sciences, that subglacial sediments had biogeochemical processes going on in them, just like freshwater and marine sediments. Now I'll hand over to Mark and let him take the story further.
Can you see the shared screen or not?
It's for full slide set, so you just need to share it.
Okay. I'm confused here. Hold up. Stop share. Share screen. I want to share my screen, which is this one. doesn't seem to want to work. Sorry, folks.
It worked last night brilliantly, Mark.
Yeah, I don't know why it's not wanting to share my screen. It says your screen sharing is paused, but I'm not pausing. Okay, why does it not want to let me share the screen? Is that now showing the screen?
It's showing the screen, but you probably just need to hit from current slide, do you, on the slideshow?
Oh, okay, right, from current slide. Do we now see it?
No.
So, excuse me. Okay. I'm not really sure what I'm doing wrong here then, because it says you are screen sharing, and I have my PowerPoint slide up. I have my PowerPoint launched.
You are sharing a blue screen with windows on it.
Right, and what happens now if I do, does this work?
That's your slides, but it's not a slideshow.
All right, okay, so now we can make it from the beginning. Is that working? Is that working now or not?
Well, we can see your slides. We can. Oh, no, we can't.
Okay. Now you can.
We can see it in the PowerPoint itself. So is it possible to just work it like that, or do you have lots of animations?
No, I should be able to do it from beginning. Why is it? I'm really confused as to why it's not. Are you not seeing it full screen?
Ernst saying go to presenter mode in PowerPoint.
Presenter mode. How do I go there?
Try clicking resume slideshow on that.
Sorry. This did work just fine yesterday. I'm just not very clear on what I should be doing to make it work.
I've got a clue here that says you can right click and go on presenter mode. So try right clicking on the slide.
Okay. Resume slideshow. Are you seeing a slideshow?
What you could do, Mark, is email it to me and I'll see if I can open it up this end.
That seems like a very smart plan, Martin. How about we do that?
There was also there was a presenter box that was unticked. It was, well, if you bring it up, I can tell you where I saw it.
It's coming through to you, Mark.
Okay.
Just a minute. No. Mark, can you just bring it up again?
Yeah.
If you see on the right-hand side,
Yeah.
it says we got monitor, primary monitor underneath. Oh, there you go.
Is this right now? Are you seeing it?
Yes.
All right, let's rock and roll then. You can see it in full screen mode?
Yes,
Yep.
we can.
Fantastic. There's a reason people don't let me use Google Drive. So anyway, right. So my apologies for that little interlude. But anyway, to follow on from what Martin was describing, I'm basically going to talk about how life is sustained in these sort of environments and then focus in on that question about pyrite oxidation. So we're looking here at a cutaway of the glacier, and we can see the area of interest is the bed of the glacier, and that is the area where the activity and the microbial activity is occurring. And in that environment, there's no light in the subglacial environment. but we do have water. We have crushed up rock, which can provide us things like iron and sulfur, nitrogen as chemical energy sources, some phosphorus as a nutrient, and then some gases, CO2, hydrogen, and methane. And we'll hear a little bit more about hydrogen from Martyn later. And then also oxygen and CO2, this could be coming out of the basal ice, could also be coming in if water is flowing in through crevasses from the superglacial environment, providing oxygenated and water from above. So in these environments, there's been microbial, or a range of microbial metabolism demonstrated in a range of systems or inferred through processes using a variety of different techniques, whether it's culturing, activity assays, geochemistry, isotope geochemistry, or 16S ribosomal RNA gene sequencing. And so, just like Martyn said, just like in marine or freshwater sediments, a range of different processes, both oxic, that can occur under both oxic and anoxic conditions. And I'm going to focus, like I'm saying, on the pyrite oxidation simply because of its importance to subglacial weathering. And so, again, to show the importance of this, this is just, this isn't meant to be a completely exhaustive list, but it's illustrative of where pyrite oxidation has been inferred or demonstrated by a variety of those different techniques to be important in driving subglacial weathering. And that's in a range of different environments: Alpine glaciers, outlet glaciers from the Greenland Ice Sheet, subglacial sediments from beneath ice streams in Antarctica. And so this process had been inferred up until about like 2010 or so, but hadn't really got a handle on like what was the magnitude of the biotic versus the abiotic. The chemistry is indicative, and there was some phylogenetic data. But this study here, published in Geology, took samples of sediments from Bodilsbreen in Norway, which is on a granitic, nice catchment, the Subglacial that we heard about before in Switzerland on some meta-sediments, and take those sediments, incubate them with the native waters, the subglacial waters, and you run one sweep abiotically, you kill it, and you run the other sweep biotically, which will give you the sum of the biotic and the abiotic. And then by difference, you can figure out like how much solid is produced in each of those incubations. And what this showed over a 100 to 300 day incubation experiment is significant release of base cations, but here back to our sulfate, so up to 20 times more sulfate being released in the biotic relative to the abiotic. And the protons that come from that sulfide oxidation producing our dilute sulfuric acid can then be used to drive the weathering of silicate and carbonate minerals to give us our sodium, potassium, magnesium, and calcium. So we're then interested in figuring out, well, if we go to the natural environment, can we demonstrate how pyrite might be involved in both looking at the microbial community in the environment, and then also digging a little bit deeper into sort of maybe the metabolisms behind driving the pyrite oxidation. So this is a sedimentary catchment up in the Canadian Rockies, small alpine glacier, Robertson Glacier, and this is sitting on top of a carbonate clastic sequence. And so in the bedrock, there's dolomitized bedrock, there's distributed pyrite, and if you focus in here, we can see all these pyrite nodules indicated by the arrows. And so the question would be, is pyrite important in determining maybe the microbial community composition? So we can test that. And so this coupon sampler approach is the brainchild of Eric Boyd, one of my longtime collaborators. And so what you can do here is you can put into these coupons, which allow water to pass through them, you crush up the minerals and you load them into the coupon sampler. So you can use minerals that have pyrite in them, so pyrite itself and the bedrock. You can use other iron-containing minerals that are oxidized, hematite, magnetite, and add in also things like quartz or calcite, things where you might not expect the microbial community necessarily to be getting energy from those particular substrates. And then you put them in the stream, put them in October, incubate them over winter, get them out in the spring, take out the minerals, extract the DNA associated with each mineral fraction, and then you can sequence the 16S ribosomal RNA out of those minerals, out of those microbial communities. And then what you can do is compare the relatedness of those communities using a phylogenetic approach. And so the relatedness of the communities, what's interesting here is that the communities that are most closely related based on the 16S work is the rock and the pyrite out of the catchment, and then water and sediment samples that were taken in October when those coupons were put in, those create a cluster. So all of the native environmental samples are most closely related to pyrite, and they're a distinct cluster from all of those other minerals. So again, that's indicating that pyrite can serve as an energy source for these microbes. So then in biogeochemistry, we're often interested in trying to link the different systems, right, in terms of thinking about, well, one of the reasons we are interested in pyrite oxidation is also that allows carbon to be fixed into the subglacial environment, right, because we've got no light. So if we want primary productivity going on, and this is again work from Eric, where you take the sediments from the environment, add a little bit of radio-labeled carbon, and then incubate them at 4 degrees C, close to in situ temperatures. And you can look at the rate at which the carbon is incorporated into biomass and see that increasing through time. One can also monitor the amount of sulfate produced and then think about the rate at which that's being produced in that reaction. And then, interestingly, if we compare that sulfate production rate from the Robertson samples or the Robertson microcosms, it's in the same kind of ballpark as from the Bodelsbreen and from the Arolla sediment microcosm experiments as well. So there used to be some agreement between these different systems, albeit in the lab experiments. So then diving into, well, what's actually driving the reactions, right, in terms of we've got microbial community, it's important. And so then thinking about the pyrite oxidation, it's not necessarily that the organisms are potentially utilizing that pyrite as a substrate by direct attachment. What we think is going on is that the thiosulfate is potentially the key intermediate. And you say, well, what's thiosulfate? Well, so if we look at this set of equations, this first equation here, the one that's highlighted here, with pyrite being oxidized to produce thiosulfate and iron oxyhydroxides. So this reaction continues abiotically, especially at pHs of about 7.5, 8, 8.5, you can get reasonable amounts of thiosulfate being produced. So this intermediate can then be utilized by microbes, either utilizing oxygen or coupling it with reduction of the oxidation using nitrate as the oxidant to then end up liberating some protons. And so then going back to our Robertson example, this is work of Sally Harold, and she was able, working with Eric and myself, to grow this particular Thiobacillus species. And this was an isolate that came out of those sediments. And so here we're looking at the aerobic growth of this organism at 5 and 15 degrees Celsius with thiosulfate. And so what's interesting is in the two panels, the left and right, one is at 5 degrees Celsius and the other at 15. If you look at the time scale on the bottom and then look at the rate at which the thiosulfate's consumed and the sulfate produced, and the cells being the cell biomass, which is the triangles, you'll see higher cell biomass and slightly more rapid rates of thiosulfate conversion at 5C than 15. Now, what this is clearly telling us is that this is an organism that's well adapted to those cold temperatures. And then one of the things that we always want to think about in these kind of studies is, well, is that isolate that we've got from the environment, is it relevant in the environment, right? You can grow numerous types of organisms from the sediments, but are they important in the environment? So going back into the environment, one can, again, this is using molecular techniques and sequencing SOX-B, and SOX-B is a protein that's in the enzyme complex that allows your thiosulfate to be oxidized. And thiobacillus, here's the sequence out of the isolate. And then all of these shaded ones, these are the ones that were extracted and amplified out of the environmental sample. So we can see the cluster that's very closely related to our isolate, and that isolate is most closely related to that environmental sampling cluster. Again, indicates it looks like it's important or relevant for the environment. So moving to slightly larger systems and thinking about subglacial environments in places like Antarctica, this is important in this context because, again, our chemo autotrophy is going to be really important because, again, in this environment, no light, but in most of these systems too, there is no sufficient input, right, between the surface and the bed. So we're going to look a little bit at what's going on in subglacial Lake Whillans, shown here with a bullseye target on this particular slide. And so this is work that was part of the WIZARD project, where we're looking at the subglacial lake and thinking about the elemental cycling of material from the lake and it draining down into the marine environment. And you'll hear more about this from Jon. So these lakes are dynamic. This is work of Matt Siegfried, showing that the lakes fill and drain on sort of decadal to subdecadal timescales, so that material from the lakes is then exported out underneath the Ross ice shelf to the ocean. And here we're focusing in on SLW or subglacial Lake Whillans. And so the microbial community in subglacial Lake Whillans, one can think about, well, is it vastly different from what you might find in these subglacial environments in, you know, Alpine and other glacial systems? And again, to orient yourself to this diagram, these are the based on 16S ribosomal RNA gene sequencing. The OTUs are operational taxonomic units. The colors with percentages. So if it says 5% OTU 2522, that's 5% roughly of the population or the sequences in that sample in the water, and 1.8 in the superficial sediments of the lake. And so what we're going to focus in on are the fact that, so very closely related to our Thiobacillus denitrificans, OTU 1767. So this is about 6% of the community in the superficial sediments. And this is an organism that would have the capability of oxidizing thiosulfate and also potentially reducing nitrate as well. And so we can also see that there are ion oxidizers, so phytotypes closely related to sideroxidans. And then we can look at the same for an ammonia oxidizer or a nitrite oxidizer, and then also a methanotrophy. And these different metabolic processes, these are all mechanisms by which the organisms can couple fixing carbon to, say, the oxidation of iron, or fixing carbon to the oxidation of ammonia. So this is a way that organisms can fix carbon in this environment. If we look at a little model of this, this is from Brent Krishna, and I've added a couple of little modifications. But so in the subglacial environment in the lake, there's oxygen coming in from basal melt and potentially the water's coming in from upstream. And that there are organisms based on the phylogeny that can be active in the nitrogen cycle. So ammonia oxidation, nitrite oxidation, nitrate reduction. Also, methane comes from deep in these environments, and the methane, we don't have direct evidence of methanogenesis in the cores, but the delta 13C and the delta D values of the methane show pretty clearly that it's microbial in source. And then we can have evidence for maybe ion oxidation and or thiosulfate oxidation. And so in these environments, clearly chemotrophy or chemosynthesis is important. And then there are also organisms that will be heterotrophs in the environment that can take advantage of this carbon that's being fixed and also legacy carbon. But if one wants to see longer term survival, you've got to have chemosynthesis, because otherwise, if it's only heterotrophy, you'll eventually run out of substrate. Anyway, I'll pass on to Jon.
Thanks, Mark. I'll see if I can share my screen. Can you see that okay?
Indeed.
Does that look all right? Okay, success, fantastic. Thanks, Mark and Martin, for providing a really nice introduction there. It's now my job to kind of do a little bit of hand waving and zoom out a little bit and talk about potentially how we can incorporate ice sheets and glaciers into global biogeochemical cycles, or at least into regional biogeochemical cycles. And I'm going to talk a little bit about my favorite element. I'm not going to reveal that yet. I'm going to build up to that and keep the excitement there. This has really motivated a lot of this work by the incredible development in our field in the last 20, 30 years, as Martin started to talk about when he did his introduction. And also the thought of ice sheets in particular as these blank spots on maps. And what I mean by that is blank spots on maps of global elemental cycles, global biogeochemical cycles. This is an example from a textbook by Milliman and Farnsworth. It's used in a lot of undergraduate courses. It's river discharge to the coastal oceans. And as you can see, the ice sheets, the Antarctic Ice Sheet and the Greenland Ice Sheet, have been left blank. This is annual discharge of dissolved solids, estimates from lots of different river catchments. And we think this is a potential issue. Why are they blank? Well, this is a young field. We haven't been working in these environments for too long, looking at geochemical things. Previously, I think a lot of people thought these environments were frozen, immobile, dead. We now know that's not true. And that's not just true for dissolved elements or dissolved solutes. It's also true for discharge of fluvial sediment to the coastal oceans. Again, the same textbook. And again, those arrows coming from these environments, even though we know that glaciers are really important agents of physical erosion. I like to call them the bulldozers of the natural environment. And so, as Martin and Mark have already eloquently pointed out, on a smaller scale perhaps, glaciers are really quite dynamic components of biogeochemical cycles. They're truncated microbial ecosystems, and they have incredibly diverse processes, biogeochemical processes occurring on the surfaces and occurring at their bed. And really the idea that we had was trying to link all these processes, the liberation of elements perhaps in these environments, and look at the impacts that might have for downstream ecosystems. So liberating elements from the rock and exporting them downstream to systems that might need some of those elements for life. And we're definitely not the only people who've done this. In the last 10, 20 years, there's really been an explosion in people looking at biogeochemical cycling, in particular, trying to link glaciers with downstream biogeochemical cycles, so the downstream cascade. Some of these ideas are still very contentious. I'm really just scratching the surface here. Really, lack of data is a big issue. There's some circumstantial evidence for sure to suggest that coastal environments around ice sheets are highly productive in areas, and there's lots of different reasons behind that. And there's been a really great set of papers that have come out to try and address some of these issues and try and address some of these questions. And this is just a handful of them. So biomass of living things contains about 30 elements. Or microbes in particular need around 30 elements to survive. Some of them are in very, very small quantities. I'm not going to talk about all 30 elements. I'm just going to talk about my favorite element for 2021, which is iron. Now, iron is a really important element in a number of ways. It's a ubiquitous element in a number of ways as well. It's found in very high concentrations in the lithosphere, 4% abundance in the upper Earth crust, roughly, give or take a few percent. It's an essential nutrient, essential element for microbes. It's an important component of cellular processes, in particular for phytoplankton, the plants of the ocean. It's involved in further synthesis, nitrogen fixation, phosphorus utilization, generation of ATP, lots of different processes, needed in quite small quantities though to build some of these components. And that makes it a really vital component of Earth system Earth systems. A big thing with iron though is that it really doesn't like to stay dissolved, which makes it quite tricky to work with and also makes it tricky for organisms to access. So when you weather iron-rich minerals, whatever that is, an iron silicate or an iron oxide, and you start to dissolve it, you produce iron 2+, ferrous iron, that very quickly reacts with oxygen, of which there's a lot in our atmosphere, and creates a precipitate. So the iron precipitates out of solution when it reacts with oxygen and creates rust. And I'm sure you're all very familiar with rust. And that means it's sparingly soluble in oxygenated environments. It's found at very low concentrations, far lower concentrations than you would expect, given its ubiquity in the Earth's crust. And it's found in vanishingly low concentrations in the ocean. And that's probably the most famous environment for iron, because productivity is thought to be limited in large regions of the ocean. Up to 50% of the world's oceans at some point during the year might be iron limited, meaning the ecosystem needs iron to be productive, to be more productive. And one of the key players in this was a guy called Jon Martyn. He published a paper in the early 90s, which was roughly called the iron hypothesis. He was also kind of semi-famous, I think, for standing up in a seminar at Woods Hole and telling everyone that if they gave him half a tanker of iron, he'd give them the next ice age. Not sure if that's true. I think it was fairly tongue-in-cheek. But he's also the guy who really discovered that iron could be a really important element in ocean ecosystems. This is me simplifying a very complex system in one slide, but it's really to frame the rest of the talk. What makes iron bioavailable? I've told you it doesn't like to stay dissolved in water, which is a big issue for organisms. There's a few things that make it more reactive to organisms, or more bioavailable to organisms. The first one is size, and that's fairly self-explanatory. Smaller particles have higher surface areas and more reactive. The second one is crystallinity, and I learned a lot of this from Leanna Benning, among others. Amorphous to crystalline particles. So amorphous particles lack structure. They have very high surface areas. They're much more reactive than crystalline particles. Crystalline particles have structure. And this is a good example here from one of Rob Raiswell's papers. These particles here, they look like little stars. These are goethite nanoparticles. And these particles here in the ovals are aggregates of a mineral called ferrihydrite, and that's an amorphous mineral, and it looks kind of like fluff almost. And the third one, and receiving increasing amounts of attention, I think, is valence state. And iron is predominantly found in two valence states at the Earth's surface: iron 2, or iron 2 plus ferrous iron, and iron 3 plus ferric iron, with ferrous iron thought to be more bioavailable than ferric iron. So the mighty Greenland Ice Sheet, this is where it all really started with me, working with Gemma Wadhams, my PhD supervisor, back in the early 2010s, I guess now. Greenland covers about 20% of land surface area in the Arctic, depending on what definition of the Arctic you use. Exports about 1,000 kilometers cubed of fresh water to the oceans every year. I don't need to tell everyone here that, but it also exports a huge amount of sediment. And really, for me, my eyes were opened from the Edinburgh group. Tom Cowden published this paper in 2012, I think it was, observing very rapid rates of erosion beneath the Greenland Ice Sheet. And then Irene Overeem published this really nice paper just a few years ago, generating some estimates of how important the Greenland Ice Sheet might be in global sediment budgets. And she was arguing that it covers about 1% of land surface area. It could be responsible for 8% plus of global sediment discharge to the oceans. This is our study site. Some people might have heard of this place before, Leverett Glacier in southwest Greenland, very close to the settlement of Kangerlussuaq. Very brief methods. Don't want to bamboozle you with lots of different terms and different methodologies. And again, this is simplifying things. So if you hark back to when I was talking about size, iron can be operationally defined in concentration depending on how you filter it, where you filter it, and what time you filter it. But I'm going to make this very simple and say that anything filterable is less than 0.45 in size. This includes a load of nanoparticles that can get through the pores of the filter. Anything that can't be filtered, it is something that we extract using the very selective chemical extraction, the scorbate extraction. You don't need to worry about that. But I'm going to call that labile particulate iron. And that's bigger than 0.45 nanometers because it's attached to sediment, but it's still highly reactive and potentially bioavailable. And it looks like this. That's that ferrihydrite that I was talking about, amorphous iron oxyhydroxide mineral, which we know to be bioavailable to a degree. So starting on the surface waters, usually observed, very dilute, contain very little sediment. It's a supraglacial lake on the Greenland Ice Sheet. Really important for driving the hydrological system, these surface waters, as you all know. I don't need to explain all of this. The evolution of the subglacial hydrological system over the season is really important for the biogeochemistry as well. But these surface meltwaters have very little iron in, really. measurements that I've taken over the years indicate this could be anywhere from about 10 nanomolar to about 30 nanomolar of iron. That's not very much. The Leverett Glacier outflow, where the meltwater comes out the base of the ice sheet, contains on average over 1,000 nanomolar of iron, up to 4,000 nanomolar of iron. So really the business end is in the bed. In the subglacial environment, where you have very high rock-water ratios, you have bedrock comminution, as Martin has been talking about, and as Mark has been talking about, redox gradients. Parts of the bed are probably anoxic. This is really great for generating high concentrations of iron because iron doesn't like oxygen. A really diverse array of iron cycling microbes. And we know that Greenland Ice Sheet is a big wetland, basically. A lot of the bed of the Greenland Ice Sheet has water at it. And that's great for generating solute, dissolved solute. And there's some circumstantial evidence that you can generate really high iron concentrations in subglacial environments as well. So very briefly, some results from outflow. This is a time series. I want to draw your attention to this figure here. This is major iron concentration. That's basically the amount of stuff, bulk stuff that's dissolved in the water. And as with a lot of glacial environments, over a melt season, you see general dilution of major ions as you pump more surface meltwater into the bed and dilute it out. Doesn't really happen with iron. It's almost the opposite. Iron starts very low concentrations at the beginning of the melt season. We have this distributed system, and get very high late in the melt season where you have very high pH, and we observe values exceeding 4,000 nanomolar. And just to remind you of that ocean concentration of generally less than 1 nanomolar, and the riverine mean concentration, global riverine mean, is about 1,000 nanomolar. So that's a lot of iron. And there's a lot of iron attached to the sediments as well, the suspended sediments, that labile particulate iron. These are the concentrations that we recorded from Leverett as well, Leverett Glacier, and mean aeolian dust value that's been generated, mostly via work from Rob Bracewell over the years. And as you can see, there's quite a bit of this labile particulate iron, this ferrihydrite. Looks like this again, just to remind you. And more work that we've done has tried to identify the valence state of some of these nanoparticles. So whether it's iron 2 or iron 3, and relating that to how bioavailable these particles might be. And it turns out that a lot of these nanoparticulate aggregates that we thought were ferrihydrite because we extracted it and measured it, could be iron 2 rich. And that's great for some microbes. They love iron 2 rich particles. It could be more bioavailable. And we published that a couple of years ago. And so that kind of takes me further downstream to a potential iceberg source. I'm not really going to talk about this very much. Rob Bracewell started a lot of this chat off in the early noughties. Icebergs seem to be an important vector of iron into the open ocean, at least in kind of a regional or local level. And that kind of particulate material that I'm talking about that was attached to the suspended sediments could also be rafted in icebergs as basal debris freeze-on. It's more complicated than that. Mark Hotwood published a really nice paper a couple of years ago suggesting that there's this non-linear loss of iron in the coastal region in icebergs as that basal debris melts very quickly off the base of the iceberg. And it's a complicated story, but there could also be an important implication there for iceberg fertilization of the Southern Ocean around Antarctica. Taking on to my last set of data, and this is tied into the SALSA project. Mark was talking a little bit about Whillans subglacial lake. This is Mercer subglacial lake, and I'm sure you've heard about this project before. And I've been very lucky to be associated with this project to drill into Mercer subglacial lake under about 1,000 meters of ice. I say the raw we, I didn't do it. I just got given some samples, drilled into this lake and recovered some samples from subglacial Lake Mercer, which we then measured for a variety of trace elements. One of them was iron. This is a normalized graph, actually, normalized to riverine mean concentrations of iron, which are about 1,000 nanomolar. And the blue dot here, this is filterable iron, so less than 0.45 microns, so stuff that gets through a filter. The Mercer subglacial lake values were up to 20,000 nanomolar, so that's more than 10 times more concentrated than the Greenland Ice Sheet meltwaters that we've previously been sampling. So to us, this suggested that these subglacial environments in Antarctica that aren't diluted from the surface and the waters have been sat there for a very long time could be enriched in iron. And this could have really big implications if some of this iron can get out into these coastal regions of the Southern Ocean around Antarctica, which are iron limited. And really, we didn't really understand much about this in the past. And hopefully this will start a new debate as to whether subglacial meltwaters from subglacial lakes reaching the coastal regions might be important in maintaining some of the productivity that we see there. And I really just want to leave you with this and just say, well, now I think we're starting to fill in some of these blank spaces. There's still a lot of debate, healthy debate, hopefully, in the community. But people are starting to build biogeochemical models that use some of these values that we're generating to have a look at whether they can model phytoplankton, chlorophyll distributions in places like the Southern Ocean. And there's definitely a lot more work to be done there. And I just wanted to give a shout out to all those ocean modelers who are doing a fine job in trying to do this. And I'm going to pass back over to Martin.
Thank you very much, John. Well, I wanted to finish off just with what I think is an exciting new development in subglacial biogeochemistry. I started off being really interested in the chemistry of glacial runoff, and I didn't really care too much about particulates in the runoff, and really more fool me. Over the last 5 years or so, I've realised that I should have paid more attention to that over the last 35 years. And the real stars for this last part of the talk are John Telling at Newcastle and Beatrice Gill-Olivas at Bristol. And they put out a paper, or John put out a paper in 2015, rock comminution as a source of hydrogen for subglacial ecosystems. And I think this is the way forward when we start to think about maintaining life under ice sheets, and maybe even how life is maintained under ice sheets in other icy parts of the universe. And the thing that got me, and I still don't really believe these results, the thing that got me is that you can't think of anything more unpalatable to eat than a grain of sand, a quartz crystal. But the crazy thing is, you know, and quartz and silicate minerals make up a lot of subglacial bedrock. The thing is, you put a glacier over the top of these silicate minerals, and quartz in this particular instance, and all quartz is a mixture of silica and oxygen bonds. When you crush the quartz, at the surface you get these silica surface-free radicals and these SiO surface-free radicals. They're ruptured SiO bonds. Now the wacky thing is, if you wet this crushed silica, the silica surface radicals split the water. They grab OH to make their surface nice and chemically stable, and they leave behind hydrogen radicals. And these are kind of super activated things that, if they bump in together, form hydrogen gas. And for me, as an old style geochemist, hydrogen is at the bottom of my EH-PH diagram. And I simply don't believe that hydrogen can be formed from crushing and wetting rock. You know, this is crazy. Even more crazily is that the Si surface-free radicals, they can grab an H from water to leave behind hydroxyl radicals, and these are kind of super reactive things in the atmosphere that keep the atmosphere clean of pollutants. You put two of those together and you form hydrogen peroxide, you know, stuff that you could bleach your hair with, and that's right at the top of my EH-PH diagram. So I've gone from having a grain of sand, crushing it by a glacier, wetting it, say, with some regelation water, and all of a sudden we've produced hydrogen. That's in the literature, hydrogen peroxide, and people are looking for that now. And abiotically, then, we've produced two extreme redox species. Now, why could this possibly be a game changer for subglacial biogeochemistry? Well, hydrogen is a really energy-rich source, energy-rich compound as far as microbes are concerned. They generate a lot of energy by catalyzing reactions that involve hydrogen. Hydrogen peroxide is a bit more tricky in that it could oxidize the cells, but potentially microbes make a living by catalyzing redox reactions. So they take a reduced compound and they oxidize it, or they take an oxidized compound and they reduce it. And all of a sudden then, in that kind of mix and mess of subglacially comminuted minerals, you've got sites where hydrogen is being generated, sites where hydrogen peroxide is being generated. And in that melange, there are all sorts of microenvironments in which a whole host of redox reactions can occur. And potentially then you maintain microbial diversity by having this big range of micro redox environments in crushed and wetted subglacial sediments. It's even possible to cleave large organic molecules into smaller molecules, which would then, large molecules are like wood. You don't like that very much for eating. Smaller compounds like carbohydrates, you like that to power your athletics. And it's also possible then to activate the large organic molecules that are found in many types of rocks into smaller bioavailable compounds that can act as biofuel. It's not just silicate grains that produce hydrogen peroxide and produce surface radicals. Sulfides, when you crush them, they produce hydrogen peroxide. So there is production of hydrogen and hydrogen peroxide under glaciers. It's food for microbial processes, and it can support potentially a wide range and diverse range of microbes. I want to pass over to Mark Skidmore just to finish, because I still really don't believe that subglacial comminution can produce hydrogen, but there's so much data out there now that it must happen. Over to you, Mark.
Yes, thanks, Martin. So yeah, just to finish off, so stimulated by John Telling's paper and thinking about hydrogen as this source, then thinking about hydrogen entropy in glacial systems. So this is Biogeochemical in Iceland, so it's on basalt. Those rocks have high iron and silica content, and as Martin mentioned, one of the other ways that you can generate hydrogen is iron 2 can react with water to also liberate some hydrogen. High dissolved stream concentrations of hydrogen at 427 nanomoles. Martin, if you want to hit, and then Robertson Glacier, we've just seen earlier, different types of rocks, low iron and silica, an order of magnitude lower stream dissolved hydrogen. Next slide, please. So then, taking again the similar approach, this is Eric Dunham's work just published in PNAS, taking sediments out of these environments, incubating them at 4 degrees C, and then adding hydrogen as a reductant, and then a range of different oxidants. And what's clear is that in both of the systems, just like Martin was saying, both of the systems, there are organisms that will utilize the hydrogen as a reductant, and that it's much about an order of magnitude greater rates of hydrogen oxidation in that local environment, i.e. where there was higher hydrogen in the environment. And it suggests that the microbial communities in those sediments from the Icelandic glacier might be better adapted to utilize the hydrogen. So full circle, there are microbes out there that will take full advantage of that hydrogen.
So some concluding remarks then. I hope we've shown that microbial processes are critical for elemental cycling in subglacial environments. We believe ice sheets are important exporters of nutrient and trace metals into the world's ocean and should be included in global geochemical cycles. And then, bonkers though it sounds, glacier erosion happens the world over, has the potential to sustain subglacial microbial life by producing these free radicals, which in turn produce hydrogen and hydrogen peroxide. And here's something, even quartz is a potential energy source. And with that, I'll leave it for any questions that you might have.
Brilliant, thank you very much indeed. Usual way of asking questions, if you put a Q or indicate on the chat, that would be brilliant. And I'll just find Facebook on my computer as well. Anyone got any questions for Martyn, Jon and Mark? I'm still finding the Facebook version. Any questions at all? Okay, here they start pouring in after you've had a few moments to relax. Joseph Grayley, do you want to go first?
Oh, do I actually say this as well as typing it? I said, Jon, I know your favorite element is iron, but in that figure you showed, which are the anomalies in Greenland and Antarctica, titanium had the biggest spike. Any thoughts?
Yeah, don't like titanium. But no. Yeah, it's interesting. A lot of the typically immobile elements like iron, titanium, aluminium seem to be pretty mobile in subglacial environments in that filtrable phase. And it's kind of interesting. I think that's really interesting. I don't really know entirely why that is, apart from this really intense comminution of the bedrock and erosion of these fresh mineral surfaces. I mean, you see that disappear further downstream for sure. And that titanium goes, especially in marine environments, it gets buried in the near coastal zone, those low salinities. So yeah, I'm not entirely sure. And actually a lot of that titanium is only in that colloidal phase. So the stuff is not truly dissolved, or very little of it is truly dissolved. It's mainly all colloidal. So it could be clays, could be titanium oxides, could be attached to some iron oxyhydroxides, could be attached to aluminium oxyhydroxides. Yeah. Up for suggestions. I don't know if Martyn or Mark have anything to add.
I think I agree with you, Jon. Yeah.
Thanks, Mark.
I can take the next question, which was asking about the relation between iron and methanogenesis and oxidation of methane. So I'm not quite sure what the question is asking, but those are separate processes. So the production of methane is from CO2 reduction in subglacial Lake Whillans based on the isotopic data, and there's also been acetoclastic methanogenesis being demonstrated in other systems, and so that, and then the oxidation of methane, that just requires oxygen, and so I don't think there's a direct link in that particular between those particular metabolisms and the iron. So I might be missing the nature of the question, but as stated, there's not necessarily a significant link between those processes.
Brilliant, thank you. Dominic, that's is asking for a paper that detailed the ruptured silicon oxide bonds releasing the hydrogen. Is that right, Dominic? Do you want to speak, or?
Yeah, if you have a look in the references in John Telling's Nature Geosciences paper, there'll be some references to those sorts of processes there, and then hopefully watch this space in the next couple of months, and there might be some other work coming out that you might find very interesting.
Sounds good. Mark Hopwood. Dominic's just said his microphone's not working. Mark, is your microphone working?
Maybe. Ah, yes, now it is. So a bit of an open question to Martyn. So this radical chemistry that you commented on in terms of H plus, H dot and H2O2, do you have any feel for kind of what the levels of these things are in these environments? Because they're quite nasty in a biological context. So are the levels high enough to be causing issues? And if so, could the cells in these environments have an unusually strong tolerance to these things?
Oh no, that's a really good question. So the concentrations are between nanomolar to micromolar. And Mark, can you help me out with the enzyme that deals with superoxide? Superoxide. It's
superoxide
sorry, my brain's being slow. But yeah, we've had a discussion about this before with people like Brent Christner as well, thinking about the ability of maybe organisms to utilize that. But again, the question might be that when one's crushing these in the experiments, maybe one produces a little higher concentration than in the natural system that might be produced, right? In terms of in an experimental setting, it may be that in the environment, if it's only nanomolar concentrations, it's probably going to be less of a challenge for microbially.
Yeah, but Brent has shown that a lot of the microorganisms have a particular enzyme, and I think I'm sorry, I'm partially dyslexic. Unless I'm using the words all the time, they just don't stick. And I'm blowed if I could remember it's something TASE. And they have relatively high concentrations of that in their cells. So potentially they have some inbuilt protection to the superoxides.
Any last questions for this team? In that case, I'm going to share next week's, hopefully successfully. No, it's just done what happened to, there we go. Yeah. Next week's talks, we have 3 early career talks from Maurice van Tiggelen, Lynn Kalasienski, and Riley Kohlberg. So hopefully see you all here next week for a really diverse range of talks. So thank you very much indeed to Martyn, Jon and Mark for a really great talk. And oh, there's something flashing there. Is there more on the chat? If there's any more questions, you can shout out because I can't see the chat at the moment while I'm sharing my screen. I'll have to find it. Lots of thank yous, basically. So thank you very much indeed. And yeah, hope to see everyone next week.
Yeah, thanks very much for coming.
Thank you all.
Bye.
Yeah.